And then the final two grants are really looking at epigenome editing. So these, again, are two different approaches to the same issue of, can we reactivate the maternal or the silent genes on the PWS, in the PWS region of chromosome 15. So as a reminder, all individuals, whether they have PWS by deletion or by UPD or by imprinting center, they all have the genes on the maternal chromosome sitting there but silenced. And so FPWR is supporting several different approaches to try to understand how we can best activate those genes and do so efficiently and specifically. So this study from Dr. Mussolino in Germany is going to be looking at one of those approaches, and that is using what they are terming designer epigenome modifiers, so activators, so A-DEMs, that are specifically targeting the PWS region. So some approaches that are being undertaken for gene activation may activate lots of genes, even genes outside of the PWS region. An advantage of this approach is that it is meant to precisely activate the PWS region. So Dr. Mussolino and his team will develop these A-DEMs to target the specific regions on chromosome 15 and test them in PWS cells in a dish in the lab. So we're excited about this study because it is, a different innovative approach to gene activation studies, looking at the chromatin right in that PWS region, and it may allow for a more specific activation of the PWS region. And in the long term, we're supporting several approaches towards gene activation, and it will be a process of seeing what makes the most sense for moving those forward into the clinic. So this is one really interesting and exciting way to do that.